Assalamu alaikum. Good evening, everyone. Welcome to our first webinar from Saudi Arabia in cooperation between Fijin Medical Company and Ritaj. And we'd like to welcome Mr. Bob Mars from US, Texas, US. It's an honor for us to have him in our webinar as the first time Saudi Arabia. Mr. Bob, he's received the ISHIM Highest Award in 2009 at the annual meeting, the Award of Honor, and Bob now is the VP of Organization Development of Beyond Clean. Welcome, Bob.
Salam alaikum, dear friends. I'm honored to join you.
You can start.
Okay. Thank you so much for allowing me to join you for this webinar, Bander and Vision Medical. I'm so excited to join you and so encouraged by the amount of attendees that we have, and I really look forward to sharing some information with you. Hopefully you will hear something new today that you have never heard before. So with that, we will start speaking about sterilization packaging quality assurance. So as we go through this presentation, I will love to talk to you about we all know as we have our sterile packaging in our hospitals or our surgery centers or wherever we may work, that it is critically important for this surgical instrumentation to be properly cleaned and sterilized before they are placed into the sterile packaging method of choice. Also, it's very, very important that sterile fields and barriers are maintained, and our packaging plays a vital role in this. And today, our program will talk about and focus on peel packs, wraps, the blue wraps, and then rigid containers. And some of you may have seen in the past where we have used even some linens in our sterile packaging, and we don't see that very much here in the US anymore, and I would assume that it's the same in your country as well. So as you think about sterile packaging basics, the systems are used for surgical instruments to, number one, prevent contamination during use. We always would like to protect the integrity of the instrumentation. Certainly we want to permit sterilization until the instrumentation is used. And then when they get to the operating room, whatever packaging method you choose, we want to make sure that it will facilitate the aseptic presentation to the sterile field. Again, these are the types of packaging we look at. And as I stated, textiles we do not use much here in the United States anymore. And I assume for you as well. So when we talk about peel pouches, we know that they're intended for small instruments, not appropriate for a large number of instruments as you package them. They can be Tyvek or paper and plastic combined. And then we always want to make sure that we're only double peel packing if these instruments or if the manufacturer says it's okay to double peel pack. And some still say it is only okay to single peel pack. So please make sure that you check with the manufacturer's instructions for use as you're using this product. And as you can see in this picture, some of our surveyors say if your instruments are not open like this, that they could be considered contaminated. So this is a point of contention here in the United States as we work with our surveyors, because many manufacturers will tell you that as long as they are not ratcheted or closed or locked, the sterilant can come into contact with all portions of the instrument. So very important, just in case a Joint Commission surveyor might mention this to you. So I always like to add pictures of devices that I find in facilities when I perform sterilization audits. And as you can tell, this packaging on the left, they have nicely folded the inner peel pouch, which is an incorrect practice. So this is something that you always want to look for to make sure that they sequentially fit inside the other pouch. When you fold them, it adds additional layers that the steam sterilant will need to penetrate, and it can impact the efficacy of your sterilization. And then the picture on the right, again, it's wonderful that they would like to protect the instruments with a towel, but this is not a recommended practice. So if you're finding this in your facilities, it's something that you want to talk to the manufacturer about and make sure that your staff and your CSSD understand that we cannot place these linen towels. The standards actually say in AAMI ST79 that we should be providing any product that is non-linting to ensure that we do not cause infections in our area. And I know this has to be true, that none of your departments are crowded and you have to place items like this in the bins. If I talk about the sterilization standards again, they have 4 items that they would like you to think about. They want to make sure that you do not crush, bend, compress, or puncture any packaging devices that you may be using. So it's very, very important that as you're packaging, you place items in storage systems to ensure that they, in fact, do not ever impact the sterility of the device. So moving forward, additional pictures that I find, and all of these are pictures that I find when I perform my audits. As you can tell, the instrument here, you literally cannot close it any more than it is closed. So likely the surveyor would pull this off the shelf and tell you that this device is not sterile. The picture on the right, if you look closely, there is a cut perfectly in this peel pack from the curette cup. And if you ever have packaging that looks like somebody has crumbled it on the shelves or in your packaging, the surveyors will usually pull this out and let you know that this is an improper way to place on the shelves. So as we move into sterilization wrap, we know that they're non-synthetic. They're often used to wrap trays within metal trays or pans. But the one thing that I really want you to think about is that when you look at them again, that you're utilizing these products and they have no tears, no rips, and no punctures. So it's very important that as a leader in your CSSD that you are conducting preventative quality assurance inspections to look and make sure that these devices are maintaining their sterilization integrity. And as you can see, a lot of the manufacturers now that make these blue wraps, they will tell you that they do not want you to stack them, because when we think about those 4 standards, do not crush, bend, compress, or puncture, as you stack them, they can be compressed. They can also cause punctures when you move them off the shelf or move them around. So it's really important to talk to the manufacturer again about the IFU to see if it is okay to stack these devices. And on the right, you can see the broken label. So this is a packaging you would like to pull off the shelf and re-sterilize. Again, when you're thinking about it, always talk to the manufacturer about the level of labeling and tape that you're using on your pouches. So any additional paper that you add to a set and additional tape will challenge the operating room and opening these devices efficiently and effectively. But also, these additional layers can again impact the efficacy of the sterilization process. And if you look on the right, there's a perfect puncture hole in the packaging. So you always want to make sure that you catch these before they enter the operating room theater. and just some additional pictures to show what quality inspections can find if you look at them to make sure that you're pulling this packaging off the shelf. So now we think about rigid container sterilization systems. We know that they're suitable for a wide range of instrument sets. They're typically constructed of rigid metal material, usually aluminum. They're durable and they're reusable. They protect your instrumentations, and one very important part is that last bullet point is that they have to be maintained. You have got to make sure that you're taking care of these sets, these containers, to ensure that they do not lose their barrier effectiveness, which they can do if you're not inspecting them properly. And again, just some examples of what I find. So this on the left is very important because this load was actually set up for a STERRAD load, and the facility had run out of STERRAD locks to place on their containers, so they used a steam lock and placed a piece of STERRAD tape over the steam indicator, which again is not a way that we should be doing that. We want to make sure we always do it. More importantly, they have covered half of the vent in the top of this container, so now the sterilant cannot penetrate this device the way that it was designed. The one on the right that I always talk about is very important. We say here that if a device is not clean, it cannot be sterilized. And if you'll notice this residual stickiness from the sticker that they have removed and the adhesive that remains, this can allow bioburden and microorganisms to adhere to it. And then bringing it to the clean side, we actually can cause our coworkers to get sick if we don't maintain these. So I always recommend to never place a label of any kind on the outside of a container because I rarely, to be honest, I rarely find them where they have been maintained properly. So it's very important that we maintain the cleanliness of these devices. And these are just additional pictures to show you what I see and what I find as I perform audits here in the United States. And again, when we think about that process of if the device is not clean, it cannot be sterilized, it's very important. If you ever consider moving from wrap to rigid containers in your departments to reduce the amount of waste, it's a great way to go. But I always remind people, the first bullet, you will never eliminate all of your waste, and they should all be used properly. But if you choose to move toward containers, think first about your high volume instrument sets, the ones that you turn around every day that you want to get back to the operating room theater. You want to think about your sets that have the highest occurrence of holes in the blue wrappers. Maybe you could containerize those, and then certainly protect your delicate instruments. Very, very important. And again, when you think about doing this, you're always thinking about the environmental impact. So the amount of debris that you may be placing in your garbage or your refuge. So it's something to really think about. So when we think about the IFU, the instruction for use, they're based on validated testing. There's no guarantee of sterility if you do not always follow the manufacturer's instruction for use. And the FDA, here in the United States, the Food and Drug Administration provided us with this amazing document that says manufacturers are required to provide you with an IFU for their device whenever they bring it into your facility. So very important. And I bring this up because here in the United States, why maintain a sterile barrier? So we have a report that came out in 1999 that says that we had 48 to 98,000 preventable infections here in the United States. And now with the updated numbers, we know that we are closer to 98,000 deaths per year in the United States because of infections that were caused in the hospital, a hospital-acquired infection. So this is something that we really want to pay attention to, and something that is very near and dear to my heart. I will share with you, there is a man here in the United States by the name of John Nance. He wrote a book called Why Hospitals Should Fly. And he talks about quality in organizations. And this book is an amazing amazing resource. Married a nurse and heard his wife, who was a nurse, talking about issues in facilities, and he compared this to challenges that they had in the airline industry here. And he goes back to an aviation accident that was the worst in the history of the world, where two jumbo jet airliners in the 1980s, one took off and actually drove through the other. And he says that we lost 1,000 souls that day from this accident because the pilot did not respect the control tower and felt that as if he was in command and could do whatever, and he took off, so these people died. And John says this is not acceptable. And if you think about this number of deaths in the United States, 98,000, he says this is like a jumbo jet airliner crashing to the earth every day of the year. And this is a huge issue. You can only imagine what would happen if this was happening. When we have an accident here in the United States, even a small accident, recently we had an accident where one person perished on an airplane, where a Southwest airline plane, the door opened and pulled a person out, and they died, and it was in the news for weeks. Yet we don't talk about this every day in the news to try to make sure we're fixing this. So this is something that I'm very, very passionate about. And we've got to learn and make sure that we're taking care of our patients and making them very safe. So we know that surgical site infections can actually feed into that, and 2 to 5% of all of our patients can get a surgical site infection, costing up to $10 billion a year annually. And I'm also hearing numbers now that we're getting closer to 20 billion. So very, very important. The role that you play in CSSD is so vital to patient care. It's so vital. So we have to make sure that we're packaging and taking care of our patients and our products. Consult with the IFU to ensure that you're properly wrapping and containing, and that you're following the device IFU. We want to make sure that you are working very diligently to always follow the IFU. So I hope you all can see here that this washer disinfector is not functioning the way that it should be working. We have a song here in the United States where the group says, "Don't go chasing waterfalls." And I think that was a perfect illustration of a waterfall on the left side of that washer disinfector. But as you know, most washer disinfectors will hold 6 full-size trays. In this picture on the left, this hospital had 12 trays in their facility with the lids still on them, and they also placed locks so the lids could not come off. So we know that the cleaning process is not being followed. As well, there is always a shelf in the middle with another sprayer arm that they removed to fit more trays. So I encourage them to let them know that this is not cleaning the way that it should. And I asked them why they did it. And they said because of the pressure that they received from the operating room theater to get instruments to them. And I said that they do not want to do this because these instruments are not clean. And again, if we go back to not clean, they're not sterile. I will move quickly because I don't want to hold you up. So also we know that an instrument set should never exceed 25 pounds. And if they do, they can impact the sterilization efficacy. And as you see again on the left, we have an instrument tray that is almost 40 pounds here in the US, which is not good. And then I always kind of laugh with my customers and say, Liz Claiborne on the right, I'm positive she does not have an IFU, so we should not be placing instruments in a set that do not have IFUs. When we sterilize these instruments, we want to make sure that we're packaging and loading the autoclave properly. Regardless of the technology, we want to make sure that we have chemical indicators and integrators on the outside for a visual inspection, but also on the inside to ensure that the sterilant has penetrated. External indicators do not indicate sterility, but they're very important to know that this product has come in contact with a device or a sterilization modality. And at my last facility, I was called by our director of our labor and delivery where they have our babies. And she told me that they used a set that was not sterile. And this broke my heart. I couldn't understand how this happened. And we had a nurse come to the CSSD who took a tray while my staff was out picking up durable medical equipment for cleaning. They took this to the labor and delivery suite and opened it and used it. So they were not familiar with these indicators. And so we did re-education and we talked to the patient and it was so we want to make sure that our users always understand how to use these products. Again, we know that they're constructed of aluminum and plastic. We have a large variety of sizes. They're reusable. They should always be cleaned in between use following the IFU. And the IFU will indicate which sterilization technology, importance of maintaining these products. We want to make sure that we inspect them to make sure that they're free of dents and pitting and anything that may damage that set. We want to make sure that the instrument set, the lid and the bottom are maintained appropriately. And if you have to pound or work really hard to open your container, this is not the way that it should be functioning. So they should always fit. We want to make sure that the filter covers are working properly, the handles function properly. And here's a good example, again, of a set that I found while doing an inspection where the edge is certainly damaged, and then you can see the filter, or excuse me, the gasket is cut and damaged. So this can impact the sterility of that device for sure. So we want to make sure that we're taking care of that. As you move to loaner instrumentation, always remember that before you stick the loaner tray into a sterilizer, you want to ensure that the manufacturer of the instruments and the manufacturer of the container have worked together to make sure that this device is validated. You want to review their IFU, position the instrument set so it will allow the sterilant into the container, eliminate water retention, and again, you should never exceed 25 pounds. Again, you're going to check for those external markers to make sure that they have changed after sterilization so that you're using a safe product for your patients. Again, lifting the lid in the operating room, the circulator will lift it. I myself am also a scrub technician, so I would always look in the container to make sure that the instruments and the indicators have changed and they have been touched by the sterilant modality that they've gone in, whether it's low temperature or high temperature, and that they have in fact changed before I pull that set out and place it on my back table where I could contaminate the whole back table. So very, very important. All containers, again, should be cleaned in between our use. We want to follow the manufacturer's IFU, and I will often joke here in the United States and say we want to follow the IFU, not the IFU, which could be a very, very bad word here in the United States. We know because they're so challenging and they're so different. I always encourage manufacturers to make the IFU as simple as we can to ensure that the users understand what they are working on and using. So we know that wrappers are composed of polypropylene. It's a melt process. They are single thickness, often thermal bonded with 2 layers. They have some new ones that require or that have different colors. We want to make sure that they're always FDA cleared. They're compatible with our technologies. We want to, again, make sure that they can, in fact, be stacked before we stack them. And what is the appropriate weight of the sets? We know it's 25 pounds. Again, as you perform your audits, you want to make sure that you're looking to make sure that if you have blue wraps on the shelf, that they are maintaining the integrity of sterilization. For when considering this, when wrapping instruments, you want to make sure you use reinforced wraps. They have tray liners out there now that you can use. They have corner protectors that will protect your instruments. You want to make sure that your shelving is protected so that you do not damage these devices as you place them onto the wrapper. And in this upper picture, you want to always make sure that you're following the manufacturer's instructions for use. This set that was sterilized in 2006, I found on the shelf in 2017. And we know that the manufacturer of the blue wrap says that you cannot leave this on your shelving for more than 10 years. It will not maintain the integrity. When they're open, we want to make sure that they're being checked properly in the operating room theater, that they are being used, that the scrub person and the circulator understand that there are no holes again, to ensure that we have the proper processes. So when you think about these devices, you always want to think about education and training your staff for principles of packaging, selection of a packaging system, making sure that you're following the IFU. You want to look at the risks and have mitigations that you can use to deal with issues as they arise. You want to have monitoring systems in place, and then action plans for when you have a breach to the sterile barrier. In summary, we today talked about surgical site infections. We talked about maintaining these instruments and sterilization, rigid containers and wraps and peel packs and the challenges related to them, and maintaining. We know that maintaining the sterile barrier is only possible when recognized guidelines for packaging the device and the instrument manufacturers' IFUs are followed. And I just want to end on a note to send a heartfelt thank you to everyone who has joined us on this webinar. But more, I would like to thank you from the bottom of my heart for what you do every day in CSSD. Do you know and do you understand that by going to your facility, you have an impact on the lives of the patients in your facility every day? You are professionals working in a department that literally the hospital would stop operating if you were not there. And what you do every day has the impact. And I take this so dear to heart. I was sharing with Bander some challenges that I've experienced. About 9 weeks ago, I had my 35th, my operation on me. I have implants in my shoulders, my hip, my back, my neck. And it's because of people like you, professionals like you who work in sterile processing in CSSD, who take care of people like me who have surgical procedures. I have never had a surgical site infection. And this is directly related to the amazing care and the amazing job that you do every day. So thank you from the bottom of my heart. It truly has been an honor for me to join you. As we end this presentation, thank you, of course, for attending, but you have my contact here if you have any questions about this presentation, or if you would like a copy of this presentation, feel free to email me at bob@beyondclean.net, and I will get this information to you. And again, thank you again from the bottom of my heart for allowing me to join you.
Thank you, Bob. Thank you so much for your presentation. Assalamu alaikum. Good afternoon. Welcome to our webinar. Today, inshallah, we will talk about how we choose for the disinfection stuff and what our measurement and factors also, and how we categorize our choice when we choose the disinfectant based on the test of devices or also indications or microbiology test. Also, we'll discuss today the quality assurance tools, what we can use after we process the instruments, and to make sure we are using the right and correct disinfection in our department. So we have this old saying from Dr. Ernst Bodley: the selection of the disinfection methods, even the sterilization, based on the instruments itself, the category for the instruments, either non-critical or semi-critical or critical. So as we see here, non-critical and for low level disinfection, and semi-critical for high level disinfection, and the critical for sterilization, and sometimes for high level disinfection. The point I start from the saying because I will go through from the bottom to explain the different kind of disinfection, and I'm sure there is some misunderstanding for using the disinfection, some procedure. So we'll go through it and we'll give you some information and some examples. Also, we'll go through the selection criteria for the disinfection if you assign to do it as a task. So in the beginning, we need to know the kinds of chemical we are using in our department. So we have the antiseptic, the chemical kill microorganism, living skin or mucous membrane. Also, we have the bactericidal, the chemical capable to killing the bacteria agent. Also, the ferricidal and fungicidal or sporicidal, capable to killing this organism, the three organisms. Also, we have the bacteriostatic, the chemical inhibit the growth of bacteria, but don't necessarily kill them. Now we'll go through to the category for the disinfection, the different level of the disinfection. We're starting for the low level disinfection. This is for, as we mentioned, for non-critical items, which contact with the intact skin, but not mucous membrane. So it's kill bacteria and fungi and some virus. It will not kill the spores as we're doing in the thermal disinfection. We have the intermediate, also level disinfection. It's killed with the vegetative bacteria, most virus, most fungi, but there is not resistant bacterial spores like what we have in the biological indicator. It's also mainly for environmental surface used on non-critical items, also that have been exposed to pathogen are not killed by low-level disinfection, like chlorine and iodine, like alphanolic, which like soap, carbolic acid. Also, it's the action with it is relatively fast acting. So that's mean when we're using the intermediate level disinfection, you can see from if you're using the indicator for doing this disinfection procedure, you can see the action for this kind of disinfection. Also, a broad spectrum related to the broad spectrum. We will go within the next disinfection, and we'll explain more about the broad spectrum. Now we are talking about the high-level disinfection. This destroying the vegetative bacteria and mycobacteria and fungi, and enveloped also and non-enveloped viruses. We can deal this with the semi-critical items, which contact with mucous membrane or broken skin, but we don't have to sterile point of use. Like OBA, we have most of us as working in CSSD or endoscopy unit, we are using OBA and parasitic acid, hydrogen peroxide, and this is some of the advantage or needs assessment for this chemical. Critical items. This is the high risk of infection items. If contaminated, things will enter the sterile tissue. That means when we use it to the procedure room inside the operating room, so that means we will classify it as a critical items. This can be treated as a high-level disinfection or sterilization based on the manufacturer guideline. Cleaning with decontamination, as we know, and we know in our policy and procedure, most of the hospitals in Saudi Arabia, we are focusing, or actually we are facing the problem when we receive the instruments in decontamination area, not moist, not cleaned, not sprayed. So the point of use in OR should be after the procedure using a chemical, okay? And we have some standards, okay, to follow. Also, we can share it with other colleagues in operating room, like what we have here. ORN, careful instruments, recommendation number 4, instruments should be kept free of gross soils during surgical procedure. We can use also an enzymatic spray at the end of the procedure or a pre-foam. Soiled instruments like the transport cart, case cart, need to be contained and confined for transportation to the soil utility room or to CSSD. For they use the case cart after the OR procedure. And cleaning with decontamination after procedure, the first step is to remove all visible and non-visible soils. Must be done, as we mentioned, according to the manufacturing guidelines. We have to check natural pH, enzymatic detergent, mechanical friction. End stage will be the thermal disinfection or mechanical washer. We have the standards. We have some standards, and I brought the most used in our guidelines here in Saudi Arabia. The AMI instruments are decontaminated within 15 to 60 minutes after use. The ORN, care of instruments, recommendation number 5, cleaning and decontamination should occur as soon as possible after the instruments are used. So here, in these 2 points, we can come up with a plan with OR staff, our colleague in OR staff, as the CSSD staff. We can suggest to them if we have a point of communication or a special room for the storing used instruments with environmental measurement, like negative pressure, like airflow, air exchange inside the room. And instead of waiting like in the corridor, that will be difficult, or sorry, will be a danger for the patients in the hospital. Also, disassembling is the most important thing. As we know, most of us here is working CSSD, so the most problem we face is usually the disassembling. So this is a disassembling of a cleaning for decontamination and the sterilization CSSD, starting from the day 1 when you receive the instruments. So everybody in the department or the end user in the other department, they should know after the use how to disassemble the instrument and send it to CSSD. So we will not sterilize it or we will not clean it before we make sure it's disassembling. Copious rinse with RO and ionizer or distilled water. Next step, disinfection. As we mentioned in the first or second slide, germicidal solution follow manufacturer guideline regarding dilution. Must come with in contact with all surfaces, must remain in wet contact for the prescribed amount, time read, the label, thermal using a mechanical WD washer, disinfector and sterilizer, washed with the cold water, then fills the chamber with the steam, heat denture protein at lower temperature if steam is present, and pressure is to achieve the temperature of 270 degrees. Goal is the steam saturation of 100%. Here we should we should work in this, especially in the decontamination area and CSSD, with either the biomedical engineering or the general maintenance, to make sure resources we have it probably according to the manufacturing guideline in any company. So when we do an installation for any machines in our department, we need to make sure and we have it proper resources for the machine. Now we're talking about selection criteria. When we assign some time to do a selection criteria to use a disinfectant, we should put in our mind some measurement or factors to follow during choosing a disinfectant. So this is based on the data base or data analysis your department, especially if you assign a quality coordinator in your department as a CSST. And I believe we should assign in every department a quality coordinator. He will follow up with if there is any problem with the consumables, if there is any problem with the test, if there is any problem. So he or she will come up with the plan to fix it in the next time. Also to share it with the colleagues. So the first thing I think to start with is the type of contaminating microorganism. So we need to define, okay, and know what we are dealing and which microorganism we are dealing in the CSST by doing a test. Each disinfectant has unique antimicrobial attributes, degree of contamination. This determines the quality of disinfectant required and time of exposure, amount of proteinous material present, a high protein-based material absorb and neutralize some chemical disinfectant, the presence of organic matter and other components such as soap may neutralize some disinfectant, the chemical nature of disinfectant. We should understand the mode of action in order to select a proper disinfectant. Concentration, quantity of disinfectant. We should choose a proper concentration and quantity that help us suited to each situation. If we dealing with the high level disinfection or with the low disinfection, we should know doing, as I mentioned, a need assessment for each area. Contact time and temperature. We need to consider this as a specs when we planning to have a disinfectants in our department. We should read about and see what's the updated about the contact time and the temperature. Sufficient time and the proper temperature must be allowed for action of the disinfectant and might depend on the degree of contamination and organic matter load. Again, we should work when we choose any disinfectant or any consumable in our CSSD, but especially the main is the disinfection, because the start point in the CSSD, it's from the decontamination area to know what kind of the disinfectant we can use. And based on the test in lab, if we can use some protein test, residual activity and effects on fabric and metal should be considered at the specific situation. Application like a temperature will pH, and the interaction with other components must be considered. Toxicity to the environment and relative safety to the people that may be exposed. So we need to make sure when we receive any new chemical, either we choose it or anyone choose it, to read out the material safety data sheet for each chemical. The last thing will be the cost. I think when we choose a disinfectant, we're looking more for the quality and the action, the proper action for this disinfectant by doing some indicators, okay, to get out the result. That's mean the disinfection is working. Here in this table, this is a classes. I put all what we talk about comparing to disinfection. procedures and the bacterial, what we have, the bacterial. Okay, so the classes of organism ranked in order of susceptibility of disinfectant. So now, as you see in the right side, this is the level of disinfection from low until high, and starting from down, the enveloped viruses like herpes or Varicella zoster virus, okay, and also vegetative bacteria, fungi like Canadia species, okay, non-enveloped viruses, mycobacteria, and bacteria with spores. Here we will deal with our, as we working in CSSD. So the main thing in our work is quality. So when we doing the disinfection procedure, either manual or automating, we should have some indicators to know how's our choice when we chose the disinfection in the beginning. So we have a different machine and the decontamination area, ultrasonic or washer disinfector, even cart washer. We need to check, and we need to have the proper indicators to test. Also, we need to make a manufacturing guideline about the temperature and the time. Also, we need to consider, is it the cleaning efficiency by doing a calibration with the proper disinfection or chemical. So most of the washer disinfector globally are open system. So you can have any chemical and make a calibration. You can also you have like advanced indicators or advanced technology to use indicators to make sure during a high level disinfection or thermal disinfection inside your washer disinfector, you are using the proper indicator. So you can use it internally inside the washer disinfector to make sure it's returning to the right color to reaching the level of the disinfection. Thank you so much. And for any information or technical support, feel free to contact us also. So now for your questions, anything you have it, you can contact me and contact Bob, and we can answer some question, Bob. It's okay?
Absolutely.
Okay.
I have been trying to answer some typing while we were going, but I'm happy to have any other questions.
Okay, there's a question here from Sorley George. What's the shelf life of the instruments?
Very good question. So this is one, again, that I always say, refer to the manufacturer's instructions for use. They will tell you how long it can remain on the shelf. In the standards, they talk about event-related versus date-related storage, and most have moved to event-related. But now, as the Joint Commission surveyors tell us to follow the manufacturer's instructions for use, they actually have a date for how long our product can remain on the shelf. And if we exceed that date, it could in fact be considered an event, and the surveyor can cite your facility for that. So always make sure that you follow the manufacturer's instructions for use.
There's a question here about from anonymous attendee. What kinds of disinfectant can be used in radiology machines? You can go back to what kind of machine they are using in the radiology department and ask them for the manufacturing data sheet. And there is a part usually for disinfection or for sterilization in the manufacturing data sheet. And you can ask the CSSD, even the CSSD, either what kind of disinfection or there is any instruments need to be sterilized. But always, as Bob mentioned, instruction for use. It's number one. When you receive any instruments or machine, you should go back to instruction for use or manufacturing data sheet. There's a question here from one of the audience, why we have to double back for OR sets and instruments?
Double packing the peel
Yes.
pouch, I assume.
Yes.
Again, this is something that is carried over from time where the operating room theater believes that you can better present this device to them aseptically if it is double packed. And it is a good way to do it. But again, I always say refer to that manufacturer's IFU, instructions for use, to ensure that you can, in fact, double peel pouch them. Some still say you may only use one pouch.
Okay. There is a question here about the 95 mask. Can be reusable, or can be disinfected or sterilized.
So here in the United States, the Food and Drug Administration, the FDA, issued an emergency authorization use for the re-sterilization of N95 masks. There is some documentation online that you can find where they talk about reprocessing it in a STERAD unit or a VPRO, and also using ultraviolet sterilization for this. Some companies have said, do not do this ever, but there are companies out there who are doing this, and it is something that we actually did here in the United States. I always share to say that it is very important that if you choose to use low temperature sterilization like hydrogen peroxide, H2O2, or STERAD, that you are off-gassing this product. So after sterilization, it has time to remove that sterilant from it, because when the nurses place this mask on, we actually are now getting complaints here in the United States that it has made some of our healthcare professionals sick. So it's very important to make sure that we're off-gassing or allowing that sterilant to have time to be removed from the device or the mask after sterilization.
There's a question about the checklist. Like most of the CSSD here in Saudi Arabia, they are using the tracking system.
Mm-hmm.
So they are asking if there is any recommendation and where we should put it, inside the set or outside the set, the container.
Yes, this is a big debate here in the United States as well. So there have been studies done on the toxicity of the printer ink that's on the count sheet. And in these studies, they have found no adverse impact to the patient. A lot of facilities here in the United States will often fold this count sheet, place it in a bag, and if it's in a container, they'll place it between the container on the inside and the basket, which is placed on top of it. We have a big issue of the ink transferring to the surgical instruments, which is never good. We don't want to damage our instruments in any way. So we have some who place it inside like that, and then we have some who have chosen to place it on the outside of the container and never place it on the inside. And honestly, most facilities seem to be moving to this practice where they're placing the count sheet usually in a bag on the handle of a sterilized set or on the outside of a blue wrapped container. So most folks here in the United States are moving away from placing them in the sterilized set.
I receive here a lot of questions regarding the sterilization of used instruments in COVID-19 patients.
Yes.
So there is any update or updated about it, or any new standards, guidelines?
Yes, so there are no new standards or guidelines. The FDA and the CDC here in the United States still recommend, in the World Health Organization, that we follow universal precautions, that we wear all of the PPE that we're supposed to wear, and we continue cleaning the devices as we clean all other devices. So our processes are set up in decontamination through the use of ultrasonic and chemistries and washer disinfectors in our sterilizers to destroy these viruses. So as long as you follow the universal precautions, you will be safe.
The most also there is too much asking about the fiber optic instruments. So they are asking about if like the laryngoscope plate be used as long as the package is intact. They are asking if it's okay, if it's intact, so they can use it, or they are following something else. As if I know, we should follow the like a production date based on the standard. And if you have something to add, you can add.
Yes, I would say as long as you remain within the date and that the package maintains its integrity, that these would be safe to use. And this is something that's very important. We used to just lay these laryngoscopes on an anesthesia counter where they were performing intubation in the operating room. And now most or all facilities have moved to packaging these laryngoscope blades. The surveyors are looking very closely at this to ensure that we're doing that. But as long as it maintains its integrity, it should be safe to use.
Yeah, also because there is some too much question about the laryngoscope because they are using nowadays with for the intubation procedure for some of the COVID-19 patients. So they were asking, can we sterilize it? Can we sterilize it? So as mentioned from Mr. Pope, we can deal with the instruments in which stage and which disease with the same by following the guidelines. Also, there is an option for the disposable plates nowadays in the market. If you, especially with the COVID-19 patients. Actually, I receive a huge number of questions, honestly. So, okay, let me. Okay, they were asking here about the environmental requirement in the sterile area. They were asking if we face a problem with the like humidity sometime. That's what I can get from the question because it's long okay. Paragraph.
Yes, the environmental standards are very, very important. So here we used to have the proper temperature and humidity to maintain contained within the AMI document, the AMI ST79. When the 2017 AMI ST79 document was updated, they removed this information from that document, and it now resides with the ASHRE, ASHRE group. And this is our facilities engineers. So they maintain proper temperature and humidity in our departments. It's very important. If you fall outside of these areas for any extended time, it would be very, very important for you to perform a risk assessment, involve your infection control practitioner, the manufacturers of your devices to ensure that they have in fact maintained sterility. And I would move forward with that risk assessment team to ensure that your devices remain sterile.
There is a question here about alcohol swab. Is it enough for otoscope sterilization for one patient to another? Usually, any kind of otoscope or any kind of medical device, you should go back to instruction for use. Any device being received in CSSD or endoscopy unit, you should, for the first time, you should ask for instruction for use. So always you will find out there is a part for the disinfection either or sterilization. Um, we have 1 minute left. I would like to thank you, Bob, so much. Actually, we appreciate your contribution in our webinar here. And for the audience also, we'd like to thank them to join us in this webinar. And we'll see you, inshallah, soon, Bob, inshallah, with the next webinar. We will plan for something because I think most of the CSSD staff here or related in infection control in Saudi Arabia, they love to be educated, get more education and training and more information, updated guidelines, standard. So thank you so much. We appreciate your time.
Thank you very, very much for allowing me to join you. It's been a true honor for me.
We too. Have a nice day. Thank you.
Thank you.